Hey everyone, welcome. Happy Pi Day and welcome to this Invasive Crayfish Collaborative webinar. My name is Natalia. I'm with Illinois Indiana Sea Grant and am the main facilitator of the Invasive Crayfish Collaborative. And for those of you who don't know what the ICC is, it's a program that brings together a variety of experts and stakeholders to address the threat of invasive crayfish in the Great Lakes region. We create monthly newsletters highlighting recent crayfish literature and news. We host webinars like this one about new crayfish research and programs. We also work and collaborate with other organizations on various research projects, and in general, just try and help promote responsible crayfish practices within consumers. So before we begin, I just really quickly, as usual, wanted to share a list of different links that you can follow if you're interested in learning more about the ICC. You can check out our main website, invasivecrayfish.org, which acts as a one-stop shop for invasive crayfish information, and it's always being updated with new information. If you want to join the ICC membership, you can follow the join link to find our subscription form, and here you'll be subscribing to our the monthly newsletters, and you'll also receive notifications about our upcoming webinars, meetings, and other events. And then if you're interested in registering for our future webinars or viewing past recorded webinars, you can follow the last two links. And then finally, you can email me at schladuk@illinois.edu if you have any questions or comments. And I believe Carter is adding these links to the chat so that you could easily access them and not have to guess how to spell my last name. All right, so one of the goals of the ICC is to ensure that information on invasive crayfish work in the Great Lakes region is being promoted and is more accessible to the public and to other crayfish stakeholders. So we're hoping that this in turn will make people more informed and will hopefully enable them to make better decisions about invasive crayfish management. So in today's webinar, we have Dusty Swedberg from the Illinois Natural History Survey here to discuss the threat of invasive crayfish on crayfish species of greatest conservation need in Illinois. Specifically, he will be discussing the habitat associations of these SGCN species and why invasive species are one of their biggest threats in Illinois. And for those who don't know Dusty, he is a scientific specialist in asticology with the Illinois Natural History Survey at the University of Illinois Urbana-Champaign's Prairie Research Institute. He earned his bachelor's degree from the University of Missouri-Columbia and his master's from Oklahoma State University. And prior to attending graduate school, Dusty spent 5 years at the Missouri Department of Conservation working with the Crayfish Crew, which sounds like an amazing group to be a part of. Thank you so much, Dusty, for being here. We really appreciate you taking the time and educating us on all the work that you and your colleagues have produced. So to everyone joining us today, we encourage you to type in any questions that you may have in either the Q&A box or in the chat at any point during the presentation. And then after Dusty is finished with his presentation, then we'll go through them together and answer them as a group. And also, as a reminder, this webinar is being recorded and it will be posted on the ICC website and on YouTube with captions. So just be mindful of that. All right, that is all that I had. Dusty, you can go ahead and begin sharing your screen and start your presentation.
All right. Correct view, wrong view.
Sorry, what?
Is it the notes view or the?
Oh, you're good. The presentation view.
Okay, good. Perfect. Just want to make sure I had the right view on there.
Nope, no problem.
Awesome. All right. Well, thank you again for having me for this talk. It's gonna be kind of a combination of a couple other talks that I've kind of given in the past at different conferences. Both of these projects were kind of a larger scale project that we did that I was actually brought here to Illinois to do, looking at just kind of a resurvey of the crayfish fauna of Illinois. So there was multiple layers of that project, but this is the part of it that I thought was most applicable to ICC and the one I could spin to make most applicable. So with that, I'll get started. And again, thank you for having me, and thank you everyone for attending. So first, I'd like to acknowledge some of the people that helped me. The funding agency this project was largely funded by IDNR, Illinois Department of Natural Resources. And then also the technicians that helped me, Billy Nixon, Kathleen, I think is here, who helped as well with some of the sampling. And then our current lab that we have here, Dr. Caitlin Bloomer, Dr. Chris Taylor, who recently retired, but still has a habit of showing up. Molly Carlson, and also Dr. Enrique Santoyo Brito, our collections manager, for all the help that he does kind of behind the scenes. So with that, I'd also like to thank the ICC for organizing this and inviting me to talk. So with that, I'll kind of jump into a quick outline here of where we're going to kind of go. So for those people who may not know, I'll kind of give a quick outline of Illinois crayfish, kind of the state of them now. Then we'll go into kind of the meat of the talk with the SGCN species, as well as linking that with some of the invasive species work, crayfish work that we've done as part of this project. And then at the end, I'll try to tie those two a little more together and more of a kind of an outlook, speculation kind of thing of where I see us, invasive crayfish kind of going, and kind of how I see SGCNs and the work we do here playing into that. So kind of getting started, Illinois, for those who don't know, is home to approximately 24 species. 7 of those are species of greatest conservation need. 4 of those 7 are considered state endangered and threatened, such as the Vaxonius indianensis. Right here is a T&E species, as well as 2 invasive species of primary concern, Vaxonius rusticus and Procambarus clarki, as you can see right there as well. Another species that is kind of on our radar that has not been documented in Illinois that's invasive is one that's been talked about here before, the marbled crayfish. Again, not been documented in Illinois yet, but still on the radar, as well as 7 primary burrowing species in Illinois, including Lacuna-Cambarus polychromatus. So when you look at the collections through the years here, Illinois is kind of lucky that crayfish have gotten some focus throughout history, a lot more than a lot of other states, especially with the state where that diversity is kind of average. From 1886, which is one of our first collections for crayfish, or records of crayfish in Illinois in our collection, to 1969, you can kind of see that most of our collections were focused near and around the University of Champaign, as well as these drainages draining the Wabash and Lower Ohio River. Most of these collections were made by Brown as part of his dissertation in the early 50s. And then when we go to 1970 through 1994, I like to consider these the Larry Page years, where he did a lot of statewide surveys for fish and crayfish as part of that, and covered a large swath of the state. Obviously, you can see a higher density here in Southern Illinois, where those SGCN species and our highest diversity occurs. In the 1995 to 2021, the Chris Taylor years, you see, again, focus down Southern Illinois with those SGCNs, as well as more focus now up here in the north and around Chicago area, mainly targeting invasive species populations. Another way to kind of view this information is just kind of these total collections per Huckade drainage. Get pretty coverage throughout the years, especially where our diversity is highest, where you'd want a large majority of your samples. Where you see that from 1995 on, again, still doing pretty good down here in Southern Illinois, as well as up here around Chicago. Kind of falls off in some of these other drainages, but not too bad. When you look a little more recently, look at the average data collection, this is kind of where we started noticing a need. A lot of our collections on average are older. For most of the state, they're between that 2001, 2005 range. So a lot of them are almost 20 years old. So we wanted to really kind of focus back in on that and make that a priority, mainly down here where we have these species of greatest conservation need, where you still see this drainage right here where most of those samples are still almost 20 years old. And that's important as we go forward because, as many of you know, when it comes to assigning conservation ranks, things like that, you need recent relevant data. And so that was kind of one of the big overarching purposes of this project, specifically the species of greatest conservation need. Part of it was to go down here and resample these populations and update our data. So when time came, we could accurately assess conservation rates and statuses. Quickly mention, again, like I said, this was part of a larger project. We did some burrowing crayfish stuff too. Sadly, if you thought hope I was presenting on that today, you'll have to stick around for another talk, maybe some other time, for Illinois anyway. This was, I just quickly mentioned, this was also the first comprehensive study on Illinois' burrowing crayfish. So previous collections were mainly not targeting burrowing species. They were just ones that they had happened to collect as bycatch when they were doing stream sampling. So this was the first one that kind of was targeting burrowing crayfish in those habitats where they normally occur or they regularly occur. The reason this also came to the forefront was there was some taxonomic uncertainty that happened around 2020 with some name changes and things kind of getting shifted around. Essentially what Illinois thought we had one species turned out to be was reclassified as 3 species and possibly a 4th. So that was kind of another reason to do the burrowing side of it, was to start to understand those ranges better and to be able to, hopefully with all the Diogenes that no longer occurs in the state, assign some of these dots up here, these yellow dots, to population, to a specific species, instead of having them still just stand as Lacuna cambaris species. So that was kind of that talk. That's all I'm going to say with burrowing crayfish for this one. Invasive crayfish, I'll get to that. So first I want to start off with the species of greatest conservation need. Illinois has, as I mentioned, has 7 species of greatest conservation need, 4 of which are considered threatened and endangered. And they include Placidus and Kentuckiensis, which occur here in the Big Creek drainage, as well as Placidus occurs at select spots along the main stem Ohio River. The other species that occurs in streams is Vaccinius indianensis, which occurs here now in the saline drainage, now mainly restricted to these rivers and streams that directly drain the Shawnee Hills. Historically, it was probably a little bit wider ranging in the saline, but its range has since reduced since agriculture practices. And the other species that I'll just kind of touch on that's kind of required a different sampling protocol was the Vaccinius lancifer, which occurs a single lake in Alexandria County. And then kind of switching gears over here to the 3 other species of greatest conservation need that are not TNEs. You have Illinoisensis and tenebrosus, which occur across the Shawnee National Forest, kind of across the Shawnee Hills area. Tenebrosus also has a couple, just a disjunct population up in Middle Fork Vermilion. So that was kind of an area we explored as well. And then standard eye is another state endemic species. Illinoisensis is also state endemic, but standard eye is kind of a state endemic as well that occurs mainly exclusively in the Little Wabash drainage. So when we look at these species, one thing that kind of jumped out to us was kind of our lack of quantitative habitat measurements. So that was one of the things that we wanted to do, was kind of start setting the baseline for these general habitat needs at a fine scale and a coarser scale. Kind of remember this picture because that is the Little Wabash River, and I'll kind of come back to that after a bit. But just know that the reason behind this was to kind of set that baseline habitat information for these species. That way, going forward, we can not only track the presence absence of these 2 species, but also relate that to general habitat conditions and track how these rivers and stuff might be changing over time as it relates to crayfish. So our methods, for this part, we wanted to do a quantitative and qualitative portion. So quantitative sampling, we did only at sites with riffle run presences where you had that continually flowing water, where we could do a quadrat kick seine. We did about 10 to 12 per reach. We did a staggered design. That's not to have as minimal influence and to sample all three areas, left bank, center, and right bank of a stream. And some of the habitat we reported at that quadrat level was substrate, course area, depth, embeddedness, and vegetated area. Substrate, we just used a modified Wentworth scale. That's pretty commonly used with crayfish as well. As course area, we split our quadrat into 4 quadrants and documented anywhere that had how many quadrants within our quadrant had course substrate. Qualitative, we split this. Qualitative was done at all sites. The time of each depended on the stream width. Our sites were defined as 10 times stream width, so it kind of varied with how much area we had to sample. Also, it depended on if we did a quantitative sample or not. And this was really done to target, species that we might not encounter in our quantitative sampling. As many of you know, all crayfish don't always occur in all habitats. So this was to target species that may occur in those pools that we missed in our quantitative sampling. A subset of all available habitat was sampled. So we made sure to, if there was undercut banks, logs, larger substrate, always made sure to hit at least a portion of all those habitats. And some of the habitat variables recorded at the reach scale was emergent macrophytes, course area, pool depth, and macrohabitat area. So proportion of pool, riffle, run, and backwater within our total reach, as well as we performed a qualitative habitat evaluation index, which is just a measure of ranking of quality habitat. So higher value, a higher index number indicates higher quality stream, lower quality. Additionally, we got some geospatial data as well. We looked at upstream landscape disturbance index, which we modified from Brown and Vivas of 2005. This basically looks at the landscape through the NLCD, National Land Cover Data Set, and assigns a value, with low values being minimally disturbed or low impact practices, and high values being high impact land use practices. And then that you add those ratios up across the drainage and multiply them by a factor. That gives you the LDI. We also did this, so we did that for the upstream portion of our reaches, as well as a local, which we looked at the catchment. We looked at slope, drainage area, total upstream drainage area, as well as catchment area, land use change, and dominant geology. So kind of our mythical analysis slide. For the quantitative habitat data, we just kind of looked at our habitat data and how that plotted against doing basic XY plots, how that plotted against our relative density at a site. And then our qualitative analysis, we did generalized linear models to look for relationships there. So kind of some quick results. We're still kind of working through some of these, but we've got enough here to present. So from 2021 to 2023, we sampled about 71 sites, which you can see here from the gray squares and gray circles here and through the standard eye. Black squares with some of the invasive sites that we sampled. They were not included in this analysis. They just got left in the figure. So kind of looking at these results, I'll start them off by breaking them up by the three stream dwelling T&E species. First, we'll look at that's known as Placidus. Placidus kind of had this relationship with LDI. It's not a, it's kind of got some wide errors on it, so not a great relationship, but kind of moment notice the trend here that, this is a trend that kind of holds up across a lot of our teenies, as you'll see. But, they seem to be occurring in these areas of high quality or low disturbed habitat. Then when we look over here on the right, these are our densities. Again, these are our quantitative. Again, as substrate size goes up, on average, you see an increase in density. As the amount of substrate area within that quadrat goes up, you again see a increase in density. And then as embeddedness goes down, as embeddedness goes, down, you see an increase in density. So that embeddedness, let's kind of think of that as amount of interstitial space that these crayfish can inhabit that's not filled in silt and things like that. Moving on to Kentuckiensis. So Kentuckiensis is kind of an interesting one. It usually co-occurs with Placidus typically. And again, we kind of see that relationship to disturbance, again, that negative relationship, whereas disturbance or more modified land use practices increases, your occurrence decreases. So again, more natural areas. The density one's a little less telling here. And that is because, again, as I mentioned, these two, Kentuckiensis and Placidus, co-occur together. Typically, where they co-occur together, Kentuckiensis is confined to pools and margins of the riffles and runs, so you don't necessarily get a lot of them in your quantitative sampling. Interestingly enough, the one site where Kentuckiensis doesn't occur, one site we sampled that Kentuckiensis did not occur with Placidus, where it occurred independently, was this site you could consider an outlier here. So that's a kind of relationship that we're kind of interested in exploring going forward as it's a relationship between two T&E species that we can find some interest in. Then looking at Indianensis, again, this is the species that's kind of now confined to those south to north draining streams from the Shawnee National Forest, the Shawnee Hills. And you see that, now we don't have LDI down here. Now it's, of course, substrate percent at that reach scale. And again, you see that increase. So as the amount of coarse substrate increases, occurrence probability or raw occurrence goes up. Real quick. So this is, as I mentioned, in the 50s, the species was collected kind of in a couple areas within the saline that wasn't where it isn't collected now. And these areas now have since become heavily agricultural dependent. So a lot of the streams now are more like drainage ditches than they are streams. So as you can imagine, a lot of silt, not a lot of coarse substrate anymore. So that relationship, kind of may help explain why they don't occur in those areas or why they were not able to persist in those areas. Then when we look at density for Indianensis with our quantitative results, again, you see that kind of similar to Placidus, that relationship where increasing substrate size and area increases density, as well as that negative relationship with embeddedness, kind of similar to what I mentioned with that siltation now because of the agricultural practices. So quickly mentioning the other T&E species, just because it is one and I need to. We did sample for Lancifer. Again, it occurs at a single location. It occurs within Horseshoe Lake historically. It had been collected, kind of lake wide, these green triangles. But recently we were only able to collect it from a single location at the causeway. This was the first time the species had been collected in, I believe, almost 20 years or maybe over 20 years. And, it's still very hard and difficult to collect, even with extensive effort. These orange squares are areas that we sampled, and we used a wide range of methods. We used baited minnow traps. microhabitat lift nets, as well as seining and dip netting. The good news is though we were only able to collect from a location, we were able to collect juvenile individuals, so there is still some reproduction, and we were able to collect Form 1 males and females. So good news is the population is persisting. Given how difficult it is to collect and how rare it still seems to be within the lake, we can't say much more than that, other than there's some issues going on in there. We're not 100% sure why. We can speculate a little bit. I believe that there is something with this system now being completely disconnected. Down here, you can see the Mississippi River. Historically, this would have been inundated during spring floods. Now that kind of doesn't happen as frequently as it used to, I'm wondering if that plays some sort of role in that. That's something also that in the future to add on to many of the projects that I'd like to explore. So hopefully we can. Quickly on the other three, just the SGCN species, Illinoisensis, that high quality habitat kind of increases that occurrence, as well as for tenebrosus, a species that occurs, Canberra species that occurs more in the headwaters of streams, usually associated, at least in the literature, with larger substrate. I've got that documented as well here, with an increase in occurrence with an increase in coarse substrate area or coarse substrate percent. One I want to kind of single out of those three is standard eye. Towards the end of the study, we kind of noticed a gap within some of our data, that this species hadn't really been sampled a lot since the early 2000s. So we wanted to make an effort to go back and try to collect the species just to check on it, and I'm glad we did. We sampled 10 historic sites represented here by the black dots, and unfortunately, we were only able to collect it from one of those 10 sites after considerable effort was put in. So this is a species that we've known the range has been kind of slowly reducing since the 1950s, mainly being what we believe is eliminated from this major trib of the little wabash. And this is concerning because this is an endemic species. It occurs nowhere else but this little wabash drainage in Illinois. So this is one we're going to be keeping an eye on going forward, and something that might need our attention going forward as well, especially given how degraded the little Wabash River is. That picture that I told you to kind of remember, I don't know if you did, that was a photo of the little Wabash. It's usually heavily incised with almost bare banks, so a lot of sun. So kind of a quick summary of that. Not surprising, coarse substrate and high quality habitat is crucial for these SGCNs. But now we have that base groundwork, so anytime we go do more monitoring, more sampling for these SGCNs, we can not only track the presence absence of these species through time, but now their density as well as the habitat that they're associated with. So if we do start seeing changes in their abundance, presence absence, we can hopefully relate that to some change within their habitat. So again, but though for this part, we did not see a lot of change in ranges or distribution for our Artini species. They seem to be holding pretty steady in their areas, with the exception of standard eye, but it's not a TNE. But these species ranges are still relatively narrow, especially for the TNEs. As I mentioned, standard eye might warrant additional protection. It's something we're kind of currently exploring. And that kind of brings us to the next portion of the talk. The habitat here is changing. So what risks do these species have from invasive species? So far do we know none of these drainages have invasive species, but that doesn't mean that they won't. So I kind of wanted to explore that a little bit, and I'll kind of tie this all together. But first, I want to talk about some of this invasive stuff we did do as part of this project. So in Illinois, invasive crayfish, rusticus was first documented at the Northern Illinois, approximately around 1973. As well, Clark eye was first documented around Chicago around 2001. Just kind of approximate, that may change a little bit. That's just what we have records for in our collection, the earliest records of each for our collection. So, other than that, we wanted to kind of explore this. And so we did some just basic sampling from that previous figure that I showed, where we had some sites kind of scattered through here just to do some monitoring. Rusticus, we recorded 2 node locations here within the Illinois River, kind of connecting the headwaters now to the southern Illinois. So we know they're occurring through here. Clarkeye, we collected it just north of its native range in Wren Lake. This was a new collection for the species here that we believe is an invasive population, as well as here at Clinton Lake. Now the Clinton Lake one, it's kind of still up in the air whether we don't think it's established, though we think it's a possibility because we have recovered a few individuals that were sent to us that were collected that were being used as fishing bait. They were dead, but they were picked up from the shoreline, as well as we retrieved a couple from snagged fishing line and trees that had dried out rusticus that we were able to ID attached to them. So are they there? Are they established? Not sure, but there is that possibility. So I included it on this map. Looking particularly at rusty crayfish through the years and kind of how they've kind of done what invasive species do in Illinois, they just kind of boom. From 73 to 84, again, this was kind of that first collection. Just kind of see some sparse, see a little bit of distribution moving around here. And just kind of sparse individual locations. Based now on our current trends, where you kind of see that whole especially that northeast section of Illinois. I mean, it'd be hard to find a segment of stream that doesn't have rusty crayfish, as well as some populations now that are moving farther south within Illinois. And I know we probably have some populations there as well. So, we got to looking at this and this got us thinking, okay, well, we've looked at these populations historically, and we haven't really revisited a lot of them. So when I started, around the time I started, Dr. Eric Larson had published this paper from Wisconsin looking at noticing some rusticus declines within some of their water bodies up there. And they have a great extensive long-term data set up there, and they noticed these trends. We were wondering if maybe we could if we would see some of those similar trends here in Illinois. Fortunately, we don't have that extensive data set that they do, but Dr. Chris Taylor and one of his colleagues back in 1996 published this paper based off some field work they did in 94 and 95, where they did go out and kind of resample these areas for rusticus, as well as sample 5 sites that they did density estimates at. So that was really what we were focused on, those 5 sites with those density estimates, kind of compare current rusticus densities as well as sympatric stream species within those 5 sites to see how it's changed. So those 5 sites were Wadham's Creek, which was at the time the westernmost extent of rusticus in Illinois, as well as Willow Creek, South Fort Kent Creek, and Beaver Creek, which was kind of at the center of the rusticus invasion area. And then Fox River down here a little farther south and east. This was a larger order stream located kind of in an urban area. So that was kind of why that one historically was chosen as well. So we tried to match our methods as close to what Dr. Taylor and his colleague did. Lucky for us, Dr. Taylor was able to assist us in the field. And so we kind of had that firsthand knowledge. So what we ended up deciding on was doing a 5 1-meter square quadrats to kind of match the 5 square meters that they, in their paper said they sampled. Then we just kind of averaged those relative densities across all 5 quadrats. At each quadrat, we recorded depth and substrate just to have that data. And then kind of moving on, looking at the results really quick. Again, we identified native and sympatric species and recorded the densities for both. So when you look at this, it's first focused on the rusticus density from 95 to 2022. for all 5 sites except for Willow Creek, you see a large increase in density. Willow Creek, you see kind of a small increase, not super large, but still notable. But all the other sites, we go from 4.2 to 18 crayfish per meter squared, from 1 to 15, from 0.4 to 4.8, 2.6 to 13.4. I mean, significant increases in densities. And then associated with that, historically, they collected at 4 sites, they collected sympatric species out of the 5. We only got it from 1, which is pretty discouraging. It's pretty much the opposite of what Dr. Larson and his colleagues were kind of noticing up in the lakes, and some of the lakes up in Wisconsin. So that was kind of discouraging now that it's pretty much completely, from these stream segments anyway, wiped out these sympatric species. Also, I will note too that it wasn't included in the analysis, but after we finished our quantitative sampling, essentially what was quantitative sampling for rusticus and for these other species, we did a qualitative sampling habitat just to make sure that we did not, or that the species was not there. And even in our qualitative sampling, we still did not detect any of the sympatric species. So it looks like from these segments anyway, that they're either completely extirpated from these segments or these reaches, or their densities are very, very low now, to the point of it's hard to detect them. Kind of a quick summary of that before I move on to the last part, trying to tie this all together. Again, rusticus does still seem to be displacing those native species, and their densities do seem to be increasing. Again, we only had 1 site where we got a sympatric species, and only 1 site where rusty crayfish density decreased. So still a significant effect from rusty crayfish within those drainages, within those stream segments. So this kind of, now I'm going to try to tie these two together. So what threat does this information tell us about our potential threat for our SGCN species? So with that, we're going to explore that a little bit. So in Illinois, none of our SGCN species in rusticus invasion areas overlap. So we're lucky there. Some of them overlap outside of Illinois. For instance, Indianensis overlaps with rusticus kind of around Louisville and Kentucky. I have not sure of any of the relationships with that, or with the Placidus and rusticus kind of overlapping of ranges. I haven't been able to find any literature on that, so if anybody knows of any, I'd gladly like to take a look at it. And then luckily for Kentuckiensis, so far within its range, it does not overlap rusty crayfish at all. Again, this is so, we don't really have a great comparison to see, well, how would rusty crayfish affect these species? So then we got to think, and then I got to think, well, is the habitat suitable? Is the habitat within that area of Illinois where these SGCN occurs, is it suitable for rusty crayfish if they were to occur there? So luckily there's been publication on that as well. Morehouse and Tobler in 2013 kind of looked at different scenarios of rusty crayfish. They looked at through MaxEnt models and modeling suitable habitat for them. They did 3 different models, I believe. They did one that was the invasive range, one that was the only the presumed native range, then another that was invasive and native. What I'm showing here is the model where they did native and invasive. And I want to kind of draw your attention right down here to where our species of greatest conservation of need occur. You do see that slightly darker area indicating that, there is suitable habitat for these species. So if they were to be introduced, the habitat is suitable for them to occur, compared to other invasive sites and native sites as well. So this was a bit of concern, but just because the habitat's right and a species gets introduced doesn't mean that it'll be able to take over or displace that crayfish. So kind of look to the literature to kind of maybe see if that always happened. And we came up with several examples. Some work that I worked on a little bit while I was at in Missouri. And I think that Dr. Jacob Westhoff and my former boss, Bob DiStefano, worked on was kind of looking at the impact of a invasive species, Hylas, in that area invading St. Francis River drainage, where Quadruncus and Peruncus, which at the time were just teeny species in Missouri, occurred. And what they found was that after the introduction of Hylas, you saw an expansion of Hylas's range within the invaded water body and a contraction of the two imperiled species. And recently, through some work by Dr. Westhoff and others, because of some of these invasions, as well as some other habitat degradation has been kind of within the St. Francis, they've been able to get these two species, Peruncus and Quadruncus, federally listed. So that's kind of exciting news, but it shows that these ranges can become contracted, or native species can be displaced by invasives. Again, I've mentioned several times with Wisconsin, I'm going to displace a native species there. And then another example from Missouri was Neglectus, displacing the cold water crayfish, Upunctus, within the 11-point drainage as well. So though it's nothing's ever certain, there is a likelihood that if these species were ever to be introduced, the habitat's good, and the literature tells us that sometimes, the more times than not, they probably have a negative effect on the native fauna. So what if? What if that were to happen? Hopefully it never does, but what if? I think you'd most likely see species declines in Placidus, Kentuckiensis, and Indianensis. Luckily, these three species are not endemic to the state, and they do have areas where it seems like, except for Kentuckiensis, they do have places where it seems like they're already co-occurring with Rusticus. So I don't know if it'd be detrimental to the population, but it wouldn't be good. One that keeps me up at night is more this relationship with standard eye. This is a population and range that already seems to be declining due to a highly degraded habitat. And so, they already have this immense pressure. If you introduce an invasive species, and an invasive species gets introduced to there and takes hold, it could be devastating for that species. So this is one that I know throughout my career I will definitely be constantly keeping an eye on, because I think it's a big risk given that it is a state endemic with such a narrow range. So kind of looking at this from a different area, kind of tied into the ICC's mission, kind of researching for this talk, came across this, that angling is increasing. There's some negatives to that. You have an increase in possible bait bucket introductions, long range introductions, spread of invasive species. There's some negatives to that, but I don't like to focus on the negatives. I like to think of the positives with this. And I believe there's an opportunity here. With new anglers coming in, now is the time to teach these new anglers of the risks of not only invasive crayfish species, but other species as well. This new flux of people, of anglers coming in of all demographics. Let's show them now and teach them now of this danger, so it just becomes common sense going forward. Also, kind of a positive is more anglers and more people participating in these recreational activities can also provide more funding for some of these aquatic invasive species management, as well as possible mitigation. And then another one that I'll mention too is citizen science. With more people on the water, more people out there, we have the opportunity to document maybe the spread of some of these invasive species or new places these species occur, maybe before they do become very highly established or already start displacing species. So we can start trying to manage them as the best we can. An instance of this, as I was preparing this talk, I just kind of pulled up my naturalist and searched kind of arrested crayfish. And it looks like last year there was a collection in the Vermilion River in Pontiac, Illinois, which we haven't, within our collection, we don't have a specimen from that area that, based on our collection, is a range increase for rusticus within the Vermilion River by approximately 20 river miles, so pretty significant increase. So that's just kind of these things that I believe citizen science can help us with. And I'm not the first to kind of have that idea. Again, Dr. Eric Larson and some of his colleagues came out with this paper where, you know, they kind of preach for these different tools to manage and detect invasive species. And this kind of requires a collaborative approach, kind of outside the box thinking from multiple disciplines with collaborations from multiple disciplines, but really also, you know, relying on that public relations to help us detect these invasive species. I mean, there's a lot more citizens out there that aren't scientists that are. So the more people we have helping us, I think the better. But again, I think this gets stressed a lot, but education outreach still remains the most cost-effective tool. It's just the most it's easier to prevent than it is to eradicate. So again, kind of tying that all together, I can mention education and information, I think, is important. Reach a wide range and diverse audience. I think right now there's no better time to do that with the increase in angler recreation, just outdoor recreation in general. I've been fortunate enough to participate in some recently participated in law enforcement training, which is nice to see. I know they've done something similar in Missouri as well. To see conservation police officers and stuff like that now taking this seriously as well. Some of these laws have been on the books for a while, but now hopefully we will have that enforcement of those issues. So the public will know that, you know, this is an issue. Again, participation from scientists and citizens is always important. And then, you know, kind of the goal, I think, was, you know, we want that knowledge and that idea that the threat of invasive species is common. That is a common knowledge to the public. You know, we don't want them like, oh, I didn't know, you know, invasive species could be that detrimental. You don't want to hear that from an angler. You want all anglers, all outdoor recreation people to understand that threat of invasive species. And I think that has to be the goal. So with that, I'll kind of leave these takeaways up here as I take questions if you want. This is kind of everything I've talked about summarized, as well as a couple quotes down there. So again, thank you all for your time today. I appreciate it. And I'll be happy to take any questions if I have time. Thank you.
Thank you so much, Dusty. Definitely agree with you. Participation from scientists and citizens is very, very crucial to stop the introduction of invasive species and invasive crayfish. So thank you again. I currently do not see any questions in the chat or in the Q&A box, but I actually had a few for you, if you don't mind. So
yeah, absolutely.
Earlier on in your presentation, you mentioned that the two species, Faxonius placidus and Faxonius kentuckiensis, you found that they co-occurred. Do you have an idea as to why they seem to be found together?
Historically, the ranges have always overlapped. So I'm assuming that's just why. You know, I mean, neither of them are introduced or anything within their drainages. I think what I found I still find most interesting about it is that what seems to be that habitat partitioning at sites where they do co-occur. You know, when we collect them together, we'd find placidus almost exclusively in riffles and runs where Kentuckiensis now would only occur in the pool habitat. But when you take it out of that area, when you take it out of those areas where they don't co-occur, where Kentuckiensis only occurs by itself, again, it's a small sample size, so we're only able to locate one site like that. Kentuckiensis almost exclusively occurred in that riffle habitat. So I don't know, there's maybe some sort of competitive exclusion going on within that area, and I don't know why they don't co-occur across that entire range. But it is definitely something that I find interesting, to say the least. Placidus is a more wide-ranging species. It is more of a, I'd say, generalist than what Kentuckiensis is. So that may have something to do with it. Placidus outside of Illinois is a pretty wide-ranging species. So that could have something to do with it as well. But again, to our knowledge, I mean, it almost has that kind of invasive crayfish kind of vibe to it, by how they're partitioning some of that habitat now, where when they don't co-occur, they don't, but we don't have any inclination that either of them are invasive within that area.
Got it. Thank you. So this next question comes from Eric Larson. He asks, on the P. clarkii carcasses at Clinton Lake, were those near the power plant outflow? There's a around 10 degrees Celsius temp gradient in that lake related to the power plant inflow versus outflow. And he was curious if the P. clarkii were picked up on the warmer side of the lake.
No, so they were picked up. Oh, I can't think of, there's kind of a old boat ramp at Clinton Lake. It's upstream. Well, I guess it is not downstream of where that flow comes in. It is upstream of that. It's a popular fishing location that is upstream of that shore fishing area. It's one of the few places that there is access there that has it, that has kind of roadside access. But no, it was upstream of that. We did try to sample downstream of that warmer flow area, but we didn't collect anything. Again, we're still kind of trying to, again, it may not be established there. These may have just been bait that somebody used that, you know, didn't get disposed of properly. So we hope there's no invasion there, but like I said, I included it because it's still on our radar and still something we're concerned with. I think the bigger question is where they come from. We don't know who sold them as bait or anything like that around there. So yeah, kind of a long way to answer your question. No, they were upstream of that inflow.
Great. Thank you so much. Are there any protections or any efforts being made to protect the aquatic systems or the habitats that hold these SGCN crayfish species? Like you mentioned the Little Wabash drainage, and how there's a lower density of Faxonius standardi. These names are difficult. I'm very sorry. The standardy populations.
Yeah, so within the Little Wabash, there may be some conservation areas. I know in some of the headwater streams there is some, I think there is some conservation areas, but as for a wide-ranging protection, drainage-wide protection, I don't believe so. There is, luckily for the other SGCNs, a lot of their remaining habitat occurs within the Shawnee National Forest. So there is some protection for those species there based on just that being kind of a public held land to the national forest. So standard eye, unfortunately, no. But with the other 3 T&Es, yeah, there are some protection. And I think it's crucial to have that because as you saw with, as you can see with Illinois, Indianensis, you know, now they only occur where we believe they occurred kind of drainage wide within that saline drainage. now they're only occurring in those streams that directly drain that fairly protected habitat of the national forest.
Great, thank you. It doesn't look like we have any other questions in the chat or in the Q&A box, but if there's somebody else that has a question, you can email me your question and I'll send it over to Dusty. Or if Dusty, you want to give out your email, that would be also helpful too. But I think that we'll wrap it up for today. That was a really great presentation. Thank you again so much, Dusty, for giving us your time and talking about the very important work that you and your colleagues are doing. And thank you to everyone in all the attendees for joining us today. I hope you all have a great rest of your day. Thank you again so much. And thanks, Dusty. Wow, we got your email in the chat.
Yes,
Perfect.
yes. Thank you again for having me. Appreciate it.
Yeah,
Thank you, everybody.
thank you. I love the
Taking
pictures
time out of
too.
the day. Thanks. Not plenty more.